Mead Steel Education Productions and Rockingham Studios presents a video read-along. Chapter 25-1 Modern Genetics, DNA, with course instructor, Mr. Ronald Jowsey.
Greetings, my fine young scientists, and welcome back. As always, please pause or rewind the video as needed during your learning. Let's get started, shall we? Chapter 25-1 Modern Genetics, DNA. Directions: read the information provided below. Highlight important information and vocabulary, and use complete sentences to answer the questions given. A. Chemical basis for genetics. It wasn't until modern times that scientists began to understand the chemical makeup of genes and chromosomes. In the 1940s and 1950s, experiments showed that genes, the basic unit of heredity, are made up of the chemical compound DNA, or deoxyribonucleic acid. DNA is a large complex molecule found in high concentrations in the nucleus of the cell. It is responsible for passing genetic information from generation to generation. DNA also controls the manufacture of enzymes. Enzymes control cellular activity. Review questions. Question 1. Genes are composed of a chemical called blank. Genes are composed of a chemical compound called DNA, or deoxyribonucleic acid. Question 2. Name two processes for which DNA is responsible. DNA is responsible for passing genetic information from generation to generation and controlling cell activities. B. Nucleotides. The DNA molecule is made up of a long chain of repeating nucleotide units. Nucleotides contain nitrogenous bases, a sugar, and a phosphate group. The four different nitrogenous bases in DNA nucleotides are adenine, A, guanine, G, cytosine, C, and thymine, T. The sugar in DNA is deoxyribose, a 5-carbon sugar. The phosphate group contains phosphorus and oxygen. Figure 25-1 is a model representation of a typical DNA nucleotide. Review questions. Question 1. The DNA molecule is a long chain of units known as blank. The DNA molecule is a long chain of units known as nucleotides. Question 2. 4 different nitrogenous bases in DNA nucleotides are blank. 4 different nitrogenous bases in DNA nucleotides are adenine, A, guanine, G, cytosine, C, and thymine, T. Question 3. In the space below, draw and label a DNA nucleotide. In your drawing, be sure to label the phosphate group, the 5-carbon sugar deoxyribose, and the nitrogen base of the DNA nucleotide. C. DNA structure. The structure of DNA was discovered in the 1950s by James Watson and Francis Crick. According to Watson and Crick, DNA molecules are shaped like a twisted ladder. The twisted ladder structure is called a double helix, figure 25-2. The DNA double helix has two strands or sides. The strands are connected at the rungs. The sides of the ladder consist of alternating sugar and phosphate molecules. The rungs are pairs of nitrogenous bases. The nitrogen bases are attached to each other by weak hydrogen bonds. The four nitrogenous bases in DNA bond, join, together in a certain way known as base pairing. According to the DNA base pairing rule, adenine, A, and thymine, T, bond together. and guanine, G, and cytosine, C, bond together. No other combinations are possible. Whenever there is A, C on one strand, it is bonded to A, G on the other strand. Wherever there is an A on one strand, there is A, T on the other strand. Review questions. Question 1. A double helix looks like a blank ladder. A double helix looks like a twisted ladder. Question 2. Nitrogen bases are attached to each other by blank. Nitrogen bases are attached to each other by weak hydrogen bonds. Question 3. According to the rule for DNA base pairing, adenine pairs only with blank, and blank pairs only with cytosine. According to the rule for DNA base pairing, adenine pairs only with thymine, and guanine pairs only with cytosine. D. DNA replication. As a result of reproduction, daughter cells are identical to parent cells. This is because during reproduction, DNA makes exact copies of itself, replicates. These exact copies are passed from the parent cell to daughter cells during cell division. Replication of DNA occurs in both mitosis and meiosis. The process begins with an untwisting of the DNA helix, figure 25-3. The two strands that make up the helix then unzip. The weak hydrogen bonds holding the nitrogenous bases together break, leaving the molecule in the form of two strands of nucleotides. Each strand is a pattern for the new nucleotide strand. Free nucleotides present in the cytoplasm pair with the free nitrogenous bases of both strands. Because the nitrogenous bases can pair only in one way, two identical DNA molecules that are identical to the original are produced. Review questions. Question 1. When DNA replicates, it makes blank. When DNA replicates, it makes exact copies of itself. Question 2. When does replication occur? Replication of DNA occurs in both mitosis and meiosis. Question 3. The diagram below represents part of a DNA molecule. Using the base pairing rule for DNA, label the missing nucleotides. Starting with the top nucleotide and working downward. C pairs with G. A pairs with T. C pairs with G. T pairs with A. G pairs with C. And G pairs with C. Question 4. Which two bases are present in equal amounts in a double-stranded DNA molecule? Of the choices shown, answer 3. Adenine and thymine are the two bases present in equal amounts in a double-stranded DNA molecule. Remembering the base pairing rules for DNA, we know that cytosine and guanine are also present in equal amounts. That ends the read-along portion of this assignment. Use what you have learned from the reading and review questions to complete the skill practice questions and chapter quiz. Don't forget to turn it in when you've finished. Until next time, thank you for watching. Be safe, make good choices, and I'll see you again soon.